Yeah. So good evening. We'll start the class for the day. It's recording. So good evening, everyone. Which slide? Oh yeah, it's the first slide. Am I audible and then my screen is visible now?
Yes,
Yes,
sir.
sir. Yes.
Audible.
So I'm Chiranjeevi Rangaraj. I'm a PhD scholar from University of Hyderabad, and I'm also a PMRF fellow. To fulfill my teaching assistantship hours, I've been associated with NPTEL for the last 2 years, and we are seeing host pathogen interaction for the last 4 weeks, and the course number is NOC 25 and BT 21, and we are in week 5 today. And so far we have seen in the last 4 weeks, we have seen history of immunology and what is cells of immunology, organs of immunology, and then organs of immune system we have seen specifically, and how there is a innate and then adaptive immunity developed in the body. So these things we have seen so far, and then we have seen some techniques how we can generate the knockout mice or knock-in mice. All those things we have seen. So today we are going to do recap on the week 5 concepts and some practice questions, and then the doubt clearing session will be followed. Okay. Overall, this session is talking about the innate immunity. Okay. So innate immunity in the sense how we have developed by birth itself. It's not developed during the lifespan. So it includes our tears. Okay, whenever we cry now, out of the tears comes, that is also a part of the innate immune system. The mucous membrane in the all over the body, wherever the hole is there in the body. So through that porous nature, like in mouth or nose or ear, we'll be having some kind of sebum secretion, or the mucus secretion will be there. For example, saliva. Saliva is a great part of the innate immunity. Skin, then the skin will secrete certain kind of sebum that is also serve as an innate immunity mechanism. Other the texture of the skin is also working as a good innate immunity thing. So that is what we are going to see why this is needed. Before that, this is one such scanning electron microgram of number of bacteria in the body. Especially it is in the gingiva. It's nothing but gums of a human mouth. See in the gums how many microbes are there. This green colors are there. Na? This all are microbes. Okay. Someone is joining now. Okay, these all are microbes. So there was a time people started to visit that we all are having microbes in throughout our body. So you people said you are from microbiology background. So other than mouth, where the bacteria plays major role in our body, which part of the body it plays a major role? Without that, it's really difficult for us to live. You told E. coli now, you did some practical. So where the E. coli is resides in our body or any other bacteria?
Intestine, sir.
Intestine. Why it is
Intestine.
important? Yes, yes, yes. Why it is important in intestine? Any idea? There are two main reasons we need uh, probiotics in the food, or we need bacteria. Do you know why? Any thoughts? What is the function of intestine?
Digestion process.
Digestion. Yes. So our bacterial system or the microbiota present in the gut or the intestine will help in digestion. Okay. That's why when any person is having any kind of stomach issue or the severe dysentery or something, doctor will give some antibiotics, because that antibiotics will kill all the bacteria. It will not discriminate whether it's a good bacteria or bad bacteria. Then the person will always give probiotics, or he will ask you to eat curd to refill the microbiota inside your digestion. This is the first thing. Second thing, you people know we cannot synthesize vitamins, right? Do people aware of that? Vitamins we can't synthesize.
Yes, sir.
So these vitamins also can be synthesized from our dietary resources through this bacteria. Okay, so that's why bacteria are really important inside the body. There are multiple bacteria there which is helpful for the body, which is not helpful for the body. So that we are going to see. So what this statement that professor talked about, there are multiple papers suggested that in body there will be n number of mycobacteria, sorry, not mycobacteria, sorry, n number of microbes will be there. That bacteria or microbes 10 times more than the number of human cells. So that is what the paper says in 1972. So around 2016, one person stated that bacteria are 1.3 times more than the number of cells, which is 37.2 trillion. So the whole body is made up of these many cells, but still the bacteria are more, which is not even equal, which is 1.3 times more than the number of the human cells. And this estimation does not include fungi, viruses, and phages in the body, because that estimation, whatever they have done, they didn't take it. But fungi, viruses, all those things are coming under the this micro things only. Okay. And if you take the genes, okay, there is a correlation. We always say the human genome has around 30,000 genes actually, okay, actively replicating. But if you see the human microbiome in the sense inside the body, how much microorganisms are there, so that consists of more than 10 lakh genes. So you can see the number of genes also more active in the body when compared with the human gene itself. So we can say that whether we are living in human body or we are living in the micro body, we don't know. So that's the one quick thing he started with. The reason we started with this thing, these many bacteria are there, which means 10 times more than the human cells are there. Like let us take a 3 crore or 4 crore or 50 crores bacteria are there in our body, or the microbes are there. We should know that what is the association between the host, that is us as a human, and then the microbe will be there in the body. So these are the images wise you can say this is not having any expression, no expression, which means the commensalism is nothing but there is no benefit to the microbe, or here the host will be benefited. So this is plus, this is not plus or not minus. Okay, no effect. So that is called commensalism. So the next kind of interaction is if this is also giving something to the host and this is also giving something to the bacteria, then we can say that is a mutualism. One such example is we are taking the vitamins from the bacteria, okay, and we are giving the nutrients to the bacteria to live inside our body, right? So that is why it is called as a mutualism between the bacteria as well as a host. And there is a parasitism, that is where the issue will come. We are saying there are multiple bacteria and fungi are there in the body. Suppose by any chance this bacteria or whatever we are talking about, if it develops or if it is affects the host body, then it is called as parasitism. So this will be benefited because it will multiply in numbers more, because we are giving the nutrients required for its growth. Then it will affect the host in such a way that it will be developing the diseased state. Okay, so that's why it is called parasitism. So To see, there are two kind of immunity we have talked about so far. That is, one is the innate, another one is acquired. So today we are talking about the innate immunity. So this innate immunity, it's not started in humans only. It started long back. So if you see, it started in 700 million years ago. Okay, so that people have seen it in the sponges. So whatever the store like receptors, complement system, phagocytosis, antimicrobial peptides, this all people have seen it in nearly 700 million years ago. So these are not something new that we are talking. Okay, then it will come to the adaptive immunity. Adaptive immunity in the sense this people have done it in some fish. That is, it has jawline. So this adaptive immunity includes MHC representation, immunoglobulins, that is nothing but antibodies, and T cell receptors, RAG, and these lymphocytes are started just 450 million years ago, and now we are having both adaptive as well as innate immunity in the human body. So the evolution of, if you see the immunity, it's not something that we started at our body only has. It has been there from the nature itself. So it started from sponge where it is 700 million years ago. Now also we have both innate as well as adaptive immunity. Okay, so if you see this image, it's really clearly tells that innate immune genes are very, very slow, in the sense the pathogens will usually evolve rapidly. The reason is this innate immunity, whatever it's not specific. Okay, so it's a non-specific manner. It's not like adaptive immunity. If it is COVID comes, then it will be specific for COVID, then it will destroy. No, this innate immunity is non-specific, so it can affect anything which is non-self. Whichever it feels that it's not self genes, then it will throw it out from the body. So many of the host restriction factors undergo the positive selection, that to resist the evading mechanism of the pathogens. So what is happening? We have so much of outnumbered microbes, and then the gene pool derived from the microbes, and then it will leads to the faster evolution of the microbes into the body. In the same way, we are also having multiple innate immunity as a first line defense against the plethora of microbes. So how it is happening, that we are going to see one by one. The first one is whenever any pathogen comes inside the body. Okay, so it has containment by an anatomic barrier. So if it is coming like in the anatomic barrier, in the sense the outside the skin and everything, then it will prevent the infection. Okay, if it is not crossing our nose, or if it's not crossing our skin, it's not crossing our eyes or something, then it will prevent the infection then and there itself. Suppose there is a fail happen, our suppose you are having any cut in your hand, or some cut or some injury already happened in your eyes or in your nose, so your body is not able to stop it. Or otherwise, if you are already sick, you are having some kind of cold or fever, so already your body is sick, so it can go through anything. So that time the failure of this first hand anatomic barrier will be stopped. So then what will happen? Then the infection will happen. Then the infection occurs. Then it leads to recognition by the performed by non-specific or broadly specific effectors. So that is what we call it as this pathogen associated molecular patterns recognized by pathogen recognition receptor. Okay, so this PRRs will be there all over in the body. That is what we are going to see. Today's class is mostly on PRRs, that is pathogen recognition receptors. So that will remove the infectious agents. Suppose that is also not able to do it, then the failure is there. Then it will recruit the effector cells. Okay, then it's recognition of PAMS activator of the effector cells. Then the removal of infectious agents. So that also not happening. Again, the fail will come. Then the adaptive immunity response, that is late, which is more than 96 hours. Then transport of antigen to the lymphoid organs, that we have seen last time. Whenever the lymph node is there, then the pathogen will be entrapped inside the lymph nodes. Then there are so much of activator cells are there. Those activator cells will act against the microbes. Then finally the removal of infectious agents. So if you see, if all failure is happening only, then finally the disease progression will be there. So you see, whenever you are getting any kind of fever or something, these many processes are going inside the body. Still, it is not able to prevent the body properly. Then it will get the not only fever. Fever is a response of this adaptive and then innate immunity. But I am trying to say, suppose if you get cold or viral infection, or let us take this mumps will come na, in children. So those all things will come because these many steps got failure. So there is no mechanism to stop it. Only then the disease development will be there. So today we are, as I already told, we are majorly focusing on innate immunity as well as the pathogen recognition receptors. The first one is innate immunity has, there are components of innate immunity. It can be the physical, it can be the mechanical, or it can be the anatomical barrier. So that is what we talked about skin. Okay, and there are something called biochemical and then the chemical barriers as well, so that we are going to see in the upcoming slide what are the biochemical and then chemical barriers that we have already seen it in the last 2 or 3 classes, how the reactive oxygen species will be synthesized, how the nitrogen oxide will be produced and everything. Then there are certain things called antimicrobial peptides or the proteins, including the mannose binding protein, that is MBP. Mannose is a carbohydrate. So these are carbohydrate binding proteins. Okay. Then there is something called C-reactive protein that we have seen it in the last week itself. C-reactive protein is really, really important. It's an indicator of an infection. Okay, that's why whenever you have any infection, doctor will suggest you to take this test that is called C-reactive protein. Then another one is a acute peptide protein. This is also synthesized by liver. Okay, and this also will be come into action. Then the serum amyloid A, alpha 1 antitrypsin, ceruloplasmin, human beta defensin that we are going to see it in the next couple of slides, and the lysozyme will be there. Lysozyme also will chop the protein or the segment into pieces. Okay, then the complement system will be there. Complement system we are not going to see it today. We'll see it in the upcoming classes, so that also I'm not keeping it here. And there are microRNAs. MicroRNAs against viruses, in the sense, microRNAs basically they, this, if this is a RNA that has 5' UTR region, then the coding region, and the 3' UTR region. So the microRNAs basically it goes and binds to 3' UTR. Okay, then it will stop the translation. Okay, so that's why it is called as a microRNAs against viruses also there. These are non-coding RNAs, we'll say. Non-coding RNA. Okay, so there are other microbial and microbiological barriers, in the sense there are multiple microbes there in our body. So that will stop the other pathogens. So those called microbial or microbiological barrier, that is we are going to see. Then cellular wise we are having more cells, almost all cells like immune cells as well as the non-immune cells. So immune cells as well as non-immune cells are there, that particularly the NK cells and then the macrophages, dendritic cells, cells expressing the PRs that sensing the products of the metabolic pathways unique to a particular class of microbes, that we will see it in the upcoming slides, not here. Okay, keep it here. Yeah. So these are the mechanical, chemical as well as the microbial barriers we can say. One is skin. Okay, so the mechanical barrier in the sense what is happening here, skin, gut, lungs, and then it can be in the eyes or nose or the oral cavity. What will happen? There is a epithelial cells are there in the body. Those are joined by the tight junction. So that forms the first layer of mechanical barrier. So skin wise, if you see, then it's had the longitudinal flow and then of air and then the fluid, because our skin is exposed to air now. So any pathogen binds to here, then it will be, if any air comes, then it will be gone. Then there are chemical barriers in the sense it has the fatty acids, then fatty acids will stop the growth of the other bacteria. Then beta defensins are there, lamellar bodies are there, and cathelicidins are there, that also stop the bacterial growth. In the microbiological things, whatever there in the microbiota in the body, so that's what I've shown in the first thing. Whenever in the gut or the oral cavity also, there are multiple microbiotas there, so that also can stop the other microbial growth. So same will go for the gut. In the gut, in the chemical base, what is there? Low pH. We know HCl is secreted in the gut, so that is not that will not allow pathogen to live. Then the enzymes like pepsins, then there are other barriers like alpha defensins, cryptidins, reg3, lacticidins, and cathelicidins also will be synthesized. Then in lungs, what is there? There is something called movement of mucus of by cilia. So that also entrap the pathogen inside the body. And then there will be something called pulmonary surfactant. So it is similar to soap. Whenever we add soap, that foam will come, then it will remove all the dirt as well as the bacteria from the drasna. So that is called pulmonary surfactant. Okay, and alpha defensin as well as cathelicidin also will be there in the lungs. Then if you see eyes and nose, as I was telling from the beginning, this tears also having the considered as chemical barrier of innate immune system. And then the nasal cilia in the nose, it will be there, and enzymes in tears, and then saliva such as the lysozyme. And in tears also we'll be having antibodies that we will in the talking in the whenever we talk about adaptive immunity. Then statins will be there, and then the beta defensins will be there. So these are the overall mechanical, chemical, and then the microbial barriers. We'll go one by one. First, we talk about the skin. So skin has the epidermis layer, and which is the outer layer composed of the multiple layers of tightly packed cells. Few pathogens can penetrate these layers, and shedding of the dead skin will be there, because what will ever happen whenever one skin layer is formed, the other skin layer will be removed. That's why the flaky things will come whenever any skin is removal is there. So due to that, any pathogen is there in the dead cells, then it will be removed by uh, the shedding of the dead skin cells. And then there is something called epidermal dendritic cells that can phagocytose the pathogen whenever it comes on the epidermis layer. And these cells are extended out among other cells of the epidermis, forming a network to intercept the invaders. So it will be there if it is hands are like this, skin is there, then any pathogen is coming like this, then the cells will be there here, then it will interrupt the pathogen, then it will be killing. So this is called epidermis dendritic cells. Okay, so there is another layer called dermis layer, which is just below the epidermis layer, so which is contains the proteins, fibers called collagen, and that gives them much more strength, and then the uh, strength to the skin, so it will not allow the microorganisms to grow. Okay, so there will be a thick layer. So it's very simple now. When you have big gate in your home, and then it is filled with multiple layers, any invader will not come. That is how it happens in the many forts. Now they will be having multiple gates, so no invaders will come inside. So in the same way, our skin also have multiple layers of this collagen fibers, so that will form the thick wall where any invaders or the microorganism or the pathogen cannot enter through the skin. Then there is something called perspiration secreted by the sweat glands. So whenever any sweat comes out of the body, that also consists the salt. So that inhibits the growth of the pathogen by drawing the water. So that leads to the osmolality change here. Whenever the high salt concentration is there, then it will remove the water from the cells. Then it will that changes due to the osmolarity. Then some antimicrobial peptides will be there, like defensins or catalycidins will be there, and then the lysozyme that destroys the cell wall of bacteria. So as I already told, we will be secreting so much of serum. Some people will be having the oil skin or everything. That is a really good thing for them because that forms a innate immunity, that forms a layer to remove the bacteria or any other pathogen from the body. So that's secreted by the sebaceous glands. So that helps in the keeping skin pliable and then less likely to break or tear. So lowers the pH of the skin that to a level inhibited to many bacteria. That's why we are not having multiple infections. So these are the layers of adaptive immunity in terms of the physical barrier. Skin we talked so far, that will stop the thing. So this is a one such example of the epidermis of the skin. So it has the stratum corneum, so which will be having the most of the textures. Then the stratum lucidum will be there, and then the stratum granulosum will be there, and then the stratum spongiosum will be there. So here I just wanted to focus on this granulosum and then the spinosum. So where there will be some watertight lipid layer will be there, that will not allow, even though anything something crosses here, there will be the lamellar bodies as well as the beta defensins in the stratum spinosum, so that will kill the pathogen or invaders. Okay, so then we talk about the mucosa surfaces. Mucosa surface started from everywhere, like inside mouth, inside nose, or the gut and everything. In the entry of genitalia in male and female, all places we have in the mucosa surfaces. So this line all body cavities open to the outside environment. Unlike the surface epidermal cells that we talked about the skin, these epithelial cells are living, okay, closely associated with the lymphoid tissues where antigens are transported via M cells. So in M cells we have seen that anti-transport mechanism. One side one will be taken, then the other side it will be given. So that is related to the epithelial cells. So epithelial cells also tightly packed to prevent the entry of the pathogens, and it often only one cell layer thick will be there. So pathogens sometimes breach the barrier. So continuous shedding of the cells. So if the one layer is there like this, then it will be removed, then another layer of cells will be formed, so it will be removed continuously. So besides producing mucous membranes also produce the lysozyme, other antimicrobial peptides, and every day an adult human can swallow, digest about 1 liter of mucus. So that is the really good example. So any human can digest how much? 1 liter of, or the swallow 1 liter of, the swallow means in that saliva only we are talking about the mucosal thing. So we can swallow about 1 liter of saliva. Okay, so this is what we have talked about in terms of the bronchial ciliated epithelial, right, in the nose and as well as the lungs we are talking about here. So that has globulate cells which secrete the mucus, helps in the trapping and then the ejecting the microbes from the body. Then the cilia will be there. The cilia, what it is, the hair like structure. So whenever any hair is there, then we put the fan, what will that, the hair will go like this and like that, so it will be dancing. So any pathogen comes, this dancing cilia, what it will do, it will entrap the, it will recognize and then it will entrap inside. Okay, and there are other blood vessels will be there. Mucus gland will secrete the mucus from the bronchial ciliated epithelial tissue. Okay, am I audible or
Yeah, yes, sir.
okay, I'll go for the next. Oh. So next is the gut epithelium. So gut epithelium will be quite different from the other epithelium. So it has the globulate cells, as we already stated that it will be synthesizing the or secreting some mucous membrane. But one such good thing in the gut epithelium we will be having, that is the Paneth cells. So Paneth cells, what it will do, it will secrete the alpha defenses and the REC3. So that will be helpful in the pathogen elimination. Okay, so macrophages also will be there near the this gut epithelium system itself. Okay, so then the lysozyme will come. So lysozyme will be having multiple work. So lysozyme, what it does, any layer of the bacteria is there, so it goes and destroy the bacteria. So we have talked about last week. There are certain bacteria so called gram positive bacteria and then gram negative bacteria. So gram negative bacteria will have thick cell wall. So this lysozyme, what it will does, then it will destroy the cellular wall system. Okay, so especially the exposed lipid bilayer will be destroyed by the lysozyme. Then the cell wall is not there means then cell will be exposed easily to the any other defense system, then it will be destroyed. So there will be pores will be there. Due to that pores, everything will be cleared. Okay, so now so far we have seen everywhere defensins, catalysidins and everything. So those all are synthesized as the pre-pro molecules. So we told that there are certain molecules in our body synthesized as pre-form or pre-pro form, then it will become pro form, then it will become active form. So what it will have, what it is, if in this case any that for example pepsinogen is synthesized as inactive, okay, and it will become pepsin. So likewise, any molecule that we talk about, that alpha defensins or beta defensins, that will be synthesized as a precursor molecule called proregion, and then with the amphoteric. Amphoteric means it will be not having both plus or minus charge. It's a neutral charge will be there. Okay, amphoteric molecule. It has both the positive and the negative elements. So amphoteric region will be there. So whenever this pro region is cut, then the amphoteric region of alpha defensins will be secreted. So the same way for the beta defensins. Then the catalysidins, there is a catalin. Then the pro region, then the cut will be there. In the reg 3 also there is no pro region, but there should be some extra region of the amino acids will be there, so that will be cut. So defensins, as we told, these are there are three subfamilies of defensins are there, which is alpha, beta, and then the eta. So are distinguished on the basis of the amino acid sequences. So each family has the members with distinct activities. Some being active against the gram-positive bacteria in this, and some are being active against the gram-negative bacteria. And some others are really specific for the fungal pathogens itself. So what it will happen, as I ordered, it's an amphoteric molecule. So it can get into either positive or the negative layer. Whatever the layer it be, it can get into it. So that is called electrostatic attraction. And then the transmembrane electric field bring the defensin into the lipid bilayer. So due to the both positive and the negative charge of the this defensins molecule, it can get into the bacterial layer. So the electric field will go this side, then it will be digesting the bacterial outside membrane. If this is destroyed, then the cellular contents will be exposed to the body, so that will be killed by the body. Then something called some more proteins called antimicrobial proteins are the peptides. So including the lactoferrin. Lactoferrin is name itself suggests that it is secreted by milk glands like so from there, what it will happen, this lactoferrin usually sequesters the iron, that is Fe2 plus. So it's a potent antiviral, antifungal, and then the antiparasitic activity towards a broad spectrum of species. So in bacteria, it has a bacteriostatic property. To counter its action, bacteria has a siderophores. Okay. So in gram-negative bacteria, it has a bactericidal activity due to its binding to the lipid A of the, which is a part of the LPS. Okay, and then azurosidin, an antimicrobacterial, and then chemotactic protein, and it inactive serine proteases. And there is something called bacterial permeability inducing protein. So this is also will induce the bacterial permeability. And vitamin B12 binding proteins also there. These all are part of antimicrobial peptides, nothing but AMPVC. Then we talked about the biochemical barriers. We have seen these things in the previous classes also. So it includes the reactive nitrogen species and then reactive oxygen species. So reactive nitrogen species include nitric oxide, nitrogen oxide, peroxynitrite, and these all are synthesis from the nitric oxide. This nitric oxide is synthesis from the arginine. Okay, then what will happen? Then in nitric oxide will react with the oxygen free radical, so that will binds to something, then it will be killing the pathogen. Then the reactive oxygen species, so it synthesizes superoxide anion, hydroxy radical, hydrogen peroxide, and the hypochlorite. This all will be synthesized due to either the NADPH oxidase or the superoxide dismutase. Okay, this we all seen in the previous classes, if I'm not wrong. So then the microbiological barriers we have seen. This microbiological barriers include there are multiple like microbiotas are there, including the firmicutes, bacteroidetes, and then actinobacteria, proteobacteria, and then the other phyla. So including it starts from mouth. There are so much of firmicutes will be there. Then the this bacteroidetes will be there in the colon more. So including the ubiquitous commensal bacteria, that is lactobacillus, and then the Clostridium species, which is called the Firmicutes, and Bacteroides fragilis, it's a Bacteroides, and then the Bifidobacterium species, which is called Actinobacteria, and Escherichia chloride, that is also a Proteobacteria. So far we have seen all the mechanical barriers or the this thing related to innate immunity. So multiple barriers are there, which is not having any kind of specific function. It has non-specific activity. Okay, and there are people started from Janway. So what they said, there is a uh, link between the innate and then adaptive immunity. So that is what we are going to see. What are the link between them? So he is the one who started in the year of 1989. About he said that there is approach in the asymptote, that is evolution and then revolution in immunology. Then there is one paper came in 1996. So they stated that this is not in the human. They said that it is in the Drosophila. There is something called the drosoventral regulatory gene cascade, that is Spaxil and then toll cactus controls the potent antifungal response in the Drosophila adults. So this is where they started thinking that this came in Cell, if I'm not wrong, in 1996 in Cell Press actually. So what they said, there are certain Drosophila adults that act against the fungal thing. So it includes the spatule, toll, and then the cactus. Okay. So how they said there is some HPs that will be there that binds to the toll receptor. Then it activates the certain things called tub P2, and then there it's from the CAT, and then the rail X complex. Then the relax will come inside. Then it starts synthesizing a certain molecule called drosomycin, and then this ataxin, and then the diptrysin. So this will go and then kill the pathogen. Okay. So then there is one more paper came in 1997. So they wrote that the human homologue of the Drosophila, that is toll protein, signals activation of the adaptive immunity. So that's where it's from the GenWave lab only. So what they said in Nature, they have written it. So they tried that there is a link between innate and then the adaptive immunity. Okay, so then in 1998, there is one more paper came. So they say that there is a defective LPS signaling in C3H and then C57, this 10 SCCR mice mutation in TLR4 gene. That is nothing but toll-like receptor gene. So that is what we are going to see how it has happened. So in the innate immune response of in Drosophila, if any fungi comes, it will be sensed by the Spaxil system. Then the toll will get activated. Then the tube and then pelli, cactus and then drosal diff genes are activated. Then it gets into the nucleus. That transcription factor will upregulate more antifungal peptides. Okay, for this only people got Nobel Prize in 2011. Okay, and he started that Professor Akira, he started to study what are the genetic studies in this Drosophila genes and how it is happening, or this toll like receptors how it is functioning. That's what he started. So if you see innate immunity in the response in Drosophila, there are cells for defense will be there is fat body cells as well as hemocytes are the phagocytic cells which can engulf the any bacteria or any pathogen per se acts as part of the immune system, and the antimicrobial peptides will be there, that is a drosomycin which is diff dependent, and diptrysin, atacin, and then sexcropin which is release dependent via IMD which is called immunodeficiency signaling pathway. And the microbial sensors will be there in the sense, if microbial sensor means any microbes comes then it will be sensed by certain chips. So here it is peptidoglycan recognition proteins, that is PGRPs. There are 13 genes involved in this which bind to the peptidoglycan of the bacterial cell wall, and then gram negative binding protein which is called GNBPs. Okay, and that recognizes the LPS as well as beta 1,3 linked glucans of the gram negative bacteria which recognize the fungi, and GNBP1 that is gram negative binding proteins 1, and then the peptidoglycan recognition protein SA which involves in the recognition of peptidoglycan from the gram positive bacteria, and GNB3 which recognizes the fungi mostly. Sorry. So these are the overall sensory system that any active serine protease called GRAS will be happening. Then this triggers the proteolytic cascade that terminates in the cleavage of the protein spatial. Then homodimerization of the spatial will takes place. Then spatial dimerization will be takes place. Then the toll receptor will activated. Then finally it stimulates the antimicrobial activity. So overall, if you see whether it is a IMD pathway or toll pathway, there is a dimerization of these things will takes place. Okay, then TLRs will get activated, then tactic kinase will get activated in some cases, then finally the dorsoventral part genes will activated, then it leads to the AMP signaling, like antimicrobial peptide signaling. The same thing will get takes for IMD pathway as well. Okay. So when you compare it with human TLR pathway or the toll pathway that they have described in the uh, Drosophila, which has quite similar levels. Okay, so that we will see what is going to happen in the upcoming slides. So in pattern recognition receptors of the host, it detects certain PAMs, that is pathogen associated molecular patterns. Okay, so what it has in our human body, there is something called TLR, that is a toll like receptors which recognize the bacteria, fungi, viruses on surface and endosomes of cells to induce pro-inflammatory cytokines and then the type 1 interferons. Okay, and there is something called retinoid induced genes, one like receptors, that is called RLRs, that also the intracellular sensors recognize the RNA of RNA viruses and induce the pro-inflammatory cytokines and type 1 interferons. And there is something called nod-like receptors which is called NLRs, and which is the intracellular sensors recognize a wide range of the PAMs to induce the pro-inflammatory cytokines and involved in the processing IL-1 family cytokines. So these are the different PRRs that we talked about, TLR, okay, and then nod receptor, okay, and then the sting receptor. These things are we have discussed. We'll go one by one. What are those going to happen? So when if you see TLR, that is toll-like receptor molecule, it has something called it always there in the membrane of the host. So it has three region in the protein. One is 18 to 25 copies of leucine rich repeat, that is called LRR, that is 20 to 25 amino acids. Then there is a single pass transmembrane domain, and one more third domain will be there which is called interleukin receptor domain. Okay, so interleukin receptor domain will be activated whenever TLR is getting activated. So the finally IL-1 pathway is getting activated whenever the TLRs are getting activated. So it is including the MyD88 family. Those things also having which are intracellular. Those also having the IL-1 receptor things. And this is a one such thing how this IL-1 receptor subfamily is also having the this TAR activation domain. Okay, and I have talked about TLR. So in our body we are having n number of TLRs starting from TLR1 to TLR9. So this will detect the certain pathogen associated molecular patterns include TLR1 and 2 can detect the triacyl lipopeptide, and TLR2 and 6 can recognize diacyl lipopeptide, and TLR4 alone can detect LPS, and TLR5 and then TLR11 can detect flagellin and then uropathogenic bacteria. So that related to the proteins. Then it comes to the TLR3, which detects only the double-stranded RNA, and TLR7 and 8 recognize imidazoquinolines and then single-stranded RNA. TLR9 recognize this CpG DNAs. So this is called cell surface, and these are endosomes. So what beauty of this TLRs? It can exist both in the outer membrane, and then it can exist in the inside the cell as well. Okay, so these are the different TLRs. Once the lecture finish, you can go back and then check where what are the ligands it can be bound to, and then what are the different cell lines it has. That monocyte cells it has, dendritic cells, mast cells, eosinophils or basophils. So number of cells are having these kind of things. And if you see in this thing, TLR10, uh, so far it found only in the human. Okay, not in the mouse. So if you see TLR 11, 12 and 13, those things are always in the mouse only. So that's what I have been talking about. This TLRs can be in the surface of the cell, and then it can be in the endosome as well. So the shoe-like structure of the TLRs will allow them to recognize the any pathogen of interaction in the body. So this TLR 7, 9 and then 3 will be the intracellular, and then the surface receptors are TLR 4, TLR 2, 6 or TLR 1, 2 and TLR 11 and then 5. So there are two different pathways it can be detected by TLR signaling. One is the MID88 dependent signaling. MID88 means it's a myeloid differentiation primary response gene 88, or TRIF independent signaling, which is called TIR, containing adapter inducing IFN beta, which is interferon beta. So this can be done by macrophages, dendritic cells, plasma dendritic cells. This will be done by only the macrophages as well as the dendritic cells itself. So we are now talking about the MID88 signaling. So whenever these things are detected by these things mean this triacyl lipopeptide, diacyl lipopeptide, LPS, flagellin, uropathogenic bacteria, this will be recognized by the MID88 pathway. So then it will form a complex called MID88 IRAK and then TRAF thing. Then it will activate the kinase, that is IK kinase complex. So whenever the NF kappa B is bound to IKB, this IKB gets phosphorylated, then it will be removed from the NF kappa B. Then NF kappa B gets into the cell, then it binds to the certain genome. Then inflammatory cytokines will be upregulated. Okay, or else IRF7 will be activated alone, then that IRF7 also goes and binds to the certain gene regulatory regions, then it synthesizes type 1 interferon in the nucleus. Then the next signaling we call TRIF dependent signaling. So whenever this TLR4 is getting activated through LPS, then TRIF complex will get activated. Then it activate the MAPK pathway. Then there also the NF kappa B gets activated. Then inflammatory cytokines will come. Or else it can activate the TBK, IK this thing. Then IRF1 will be activated. Then that also comes to this IRF3. So what are the two things it's activated this TRIF signaling? That is TLR4 and TLR3. So TLR4 can use both MID dependent as well as TRIF pathway. But whereas TLR3 can activate only TRIF pathway. So this is what the overall thing, except TLR3 uh, that only uses the TRIF pathway, whereas TLR4 can be used by both MID81 and then the TRIF pathway, and most of the TLR7 and 8 and 9 and 11 and 12 and 13 will be using only the MID88 pathway. So once it detects, then it will synthesize the type 1 and then type 2 interferons. Then that can activate the vasoactive amines such as the histamine, serotonin, or eicosanoids, thromboxanes, leukotrienes, prostaglandins, or the peptide like bradykinin, and proteins like IL-1 beta, IL-6, TNF alpha. Then type 1 interferons such as IFN alpha, and then consists of 13 members, and then the signal IFN beta, and the type 1 interferon inducible genes such as IP10, RANTES will be activated, or type 3 interferons things such as IFN lambda consist of which is 3 members. So how this type 1 interferons in antiviral response, whenever the type 1 interferons are activated, then it can activate the oligoadenylate synthetase, then it will convert the ATP to 2 comma 2 prime 5 prime linked oligomers, so that can activate the endoribonuclease RNA cell, so that can chop the viral RNA. Or else type 1 interference also activate the P1 kinases, that will activate the elongation factor 2, so that will stop the translation of the proteins. Or it can also activate the MH protein, which increases the resistance to virus such as influenza. And type 1 interferons activate the NK cells, that also kill virally infected cells in the body. And it can also activate the adaptive immunity, that increase the antibody production, and then the CD8 cell response, and then the increase in MH1 MHC1, that is a major histocompatibility complex 1 expression as well. So whenever any polymorphism of TLR signaling happens, that can leads to the human disease, as well as it can help in the body. So here you see any TLR4 polymorphism, that is D299G, which is aspartic acid to glycine, that will lead to the decreased response to the inhaled LPS, then decreased atherosclerosis, increased gram-negative bacterial infections, increased severity of atopy, then the decreased airway hyperreactivity. Then whenever the IRAK4 stop codon, that is 28,287,293 is there, then it's increased the gram-positive infection. The reason is these things are not there, then the bacteria will not be recognized by the body. Finally, it leads to the disease development. Then one more thing called CD14. If that has this cytosine to threonine polymorphism in the promoter region, then it increased response to the LPS. So it's a good response in cases. And we move several polymorphism which increase the viral and then bacterial infection. Then if it is having IAKB alpha, that is to serine to isoleucine, then it will impair the T cell memory. So these are the different polymorphism. That's why when COVID also got so many people, we have seen that some people are really restricted to the pathogen, and some people are really more viral infection were there when compared with the other people. And these are the other polymorphism that we can talk, such as the TLR2, which is having T972C polymorphism, or R7532Q polymorphism. So that leads to uh, protective effect against leprosy, and then predisposition to tuberculosis, and predisposition to staphylococcus infection. And TLR4 that we have seen so far, it leads to the inflammatory syndrome, septic shock, decreased risk of atherosclerosis, no association with susceptibility to rheumatoid arthritis. And TLR5, it is there is a one dominant negative stop codon mutation that leads to increased susceptibility to pneumonia caused by Legionella pneumophila, are negatively correlated with Crohn's disease. Crohn's is a bowel disease, so that is also leads to that thing. Then if it is having any kind of mal protein or IRF5, IRAK, MID88, these all are related to this signaling of TLR. So that is also having multiple things. So here in the case, it's having the protective against the developing tuberculosis, severe sepsis, malaria or SLE. And SNP leads to increased susceptibility to SLE, that is something with systematic lupus erythematosus. Then the recurrent Staphylococcus pneumonia infection. Then here in the MID88 region, then there will be more decreased antibody response to measles. Then people have seen whether these things are having any kind of role in the therapy. So that is what we are seeing from the beginning. What are the therapies that we can give? One such great scientist, that is Coley. He called as a father of immunotherapy or the inventor of Coley's toxin. So what he did in 1891, he injected the streptococcal in patient with the inoperable cancer. So they don't have any other option to for the cancer treatment. So they have taken, he injected this thing to around 1,000 patients. So what happened? There something called shrinkage of malignant tumor will happen. That whatever the reduction in the inflammation of the or the tumor development was reduced. So this he did over the next 40 years as the head of the bone tumor service at the Memorial Hospital in New York. Cole injected more than 1,000 cancer patients with bacteria or bacterial products. So these bacteria or bacterial products were known as the Cole's toxin or used in the treatment of bone and soft tissue sarcomas. So this is the one of the drawing of this Cole's first bone sarcoma case treated with this toxins. Okay. So, but he didn't have the scientific evidence or scientific reason to say why it is happening. Then people found out that there are certain active molecules such as this OM174, which activates this TLR2 and then the TLR4 agonist. So that will help as a the cancer therapy. So this is one such thing. This OM 174 is there in that bacterial thing. Then when it is activates the TLR2 and TLR4 pathway, then it will remove the much of the cells from the body from that position. Then it leads to the reduction in the cancer. So now we are going to see what are the different compounds that have been in trial to say that these things are having the this can be helped in the cancer treatment or not. So you can see that it starts from TLR3 target, TLR2 target. That drug class is double stranded RNA molecule. Autoclaved mycobacteria also will be used here. TLR3, which is the double stranded RNA mimic. TLR4 molecule, the CBLB502. That drug class is flagellin will be there. These all are in the preclinical state. And in the phase 1, TLR9 related things are there. TLR7, this ANA 773 is there, and TLR2 related things are there, which is a lipid A derivative, and TLR9 thing is short DNA oligonucleotide things, and TLR9 is again that is agatolimod, that's also from the Pfizer. These people also done some vaccine against the COVID, so that is also in the phase 2 clinical trial. Then TLR7 against poly TLR is also there. So these all are in phase 1 and phase 2 are the preclinical stages of immune system. So wait 1 minute. I will go and take my laptop charge and come. 2 minutes. I will come. Yeah, these things are in the clinical trial. Then this is one more molecule that is imiquimod. It's a toll-like receptor 7 agonist, and it's also launched for the papilloma virus infection. Okay, And these are all are things, something. Yeah, sorry. What happened?
Voice not audible, sir.
No,
Still not
sorry. Mine has
discussed.
got got turned off. I'm just turning it on one second.
Yes, sir. Okay, sir. Okay, sir.
Okay.
Yeah, am I audible now?
Yes, sir.
Sorry. I don't know because I've turned on both this video and then this one. No? So much of battery concern actually. Yeah, I will be taking more than 10, not more than 10 minutes. Okay, we will wind it up.
Okay.
Yeah. So there are some of the molecules that like this TAK242 like molecules they have used as a TLR4 antagonist, but it has been discontinued in the recent days. And then this eritoran is a TLR4 antagonist. Antagonist it stops the TLR4 activity. That also in the phase 3 trials for sepsis. And there are other this AV42 is a TLR4 antagonist used in the ocular inflammation, which means eyes inflammation. So people nowadays they have seen multiple things are there, whether it is in the preclinical stage or in the phase one or other stages. People are trying to do it. Okay, so the not only for that, the development status of other components we see target the other TLRs because it's related to the inflammation pathway and then allergy as well, right? So if they wanted to give any drug against the allergy, asthma, or autoimmunity, that also people are trying it in the day-to-day thing. So this is what we have in the case of um,
Ten.
subject. So we'll get into some question and answers. Okay. So what's the answer for this? Microbes in the human body outnumber our own cells by how many times? Any idea? Microbes in the human body outnumber our own cells by how many times? That's the first slide that I have taken. Any idea? The people there? Am I audible? Should I tell or you'll tell? Any idea?
10.
Yes, 10 times. Good. So it's 10 times, including all virus and everything that if we talk, then it's around 10 times. Which of the following is not part of the innate immunity? TLRs, RLRs, NLRs, complement system, mRNAs. Antibodies are part of innate immunity or what?
Innate immune cell required.
Antibodies are not part of innate immunity. Okay. So this TLR, RLR, complement system, mRNA, antimicrobial peptides are part of the immune system. The one is not part, that is antibodies. That's the correct answer. Lactoferrin inhibits microbial growth by which one? Which of the options correct?
Sequencing
Endopeptidase
FE2
activity, ROS and RNS production, innate immune cells recruitment, sequestering Fe2 plus ions, induced permeability in bacterial membranes. Any idea? Lactoferrin inhibits microbial growth by
innate immune cell required.
that's it. No. Lactoferrin, it's a ferrin. What is ferrin? Ferrin. I'm giving the idea itself. Ferrin. Ferric chloride. What is ferric chloride? You people studied na ferric chloride, ferrous chloride.
Sequencing Fe2 plus ions.
Sequestering Fe2 plus ion. That's why it's called lactoferrin.
Body protection.
Lacto means it's secretion in the milk. Okay. Mother's milk will be there na, so it's secreted in the mother's milk. So lactoferrin. Okay. Which of the following is a microbial sensor in Drosophila? So that is GNBPs. Okay, gram-negative binding proteins, bacteria proteins. And which one is incorrect? That is PRR PAM interaction can, which initiate the inflammatory response. Yes, that is correct. A, because that is giving IL-1 response. And which activate innate immune response. Yes, that is also correct. Which induce the type 1 and type 3 interference. That is also correct. Initiate the cancer signaling cascade. Initiate the antibody production. This is wrong because it is not the part of innate immunity system. Okay, PAMs are unique to each pathogen, found only in adaptive immunity. It's not in adaptive immunity because PAM are related to pathogen associated molecular patterns. So responsible for creating antibodies? No, it's our innate immunity related things. Conserved molecular patterns on pathogens. Okay, it's not only present on virus, it's present in bacteria, fungi, and everything. So the answer is conserved molecular patterns on pathogens. Okay, Collis toxin, which was injected in cancer patient, was made up of. Which is the answer correct? Cholecystocin, the toxin that we have seen it in the last. Which one is correct? Tumor killing viruses,
and
bacteria, bacterial products, bacteriophage. I'll go back. Read it and tell me. It's there, no? This guy. What he did? Here you see. With bacteria and bacterial products. Right? So what he did, that guy, he took some streptococcus bacteria and he put it on the cancer patients who was terminally ill. There is no possibility. There is no possibility to remove the tumor. So then he gave this thing. So the answer is what? A, B, or C, or D, or E?
Correct.
Which one is the answer? Option?
Bacteria and
Bacterial products. Option B. So he has taken the streptococcus. So if you in any mast cell is there, then what he did? He injected the bacterial products to remove. Okay. And which among the following statements are not correct? So okay, microtimes are 10 times more than the number of human cells. Is it correct? First statement. That's the first question I asked. Is it correct statement? Yes, it's correct. Third statement. Every day an adult human swallow and digest about 1 liter of mucus. Right? You swallow? Yes, right. So that's also correct. So the wrong option is microbiome present in the human body does not include fungi. That is the wrong option. It's include fungi. All microbiome means it is a microbiome of everything, bacteria, virus, fungi, and everything. So this is not the correct statement. Which of the following statement regarding innate immunity is wrong? Okay, innate immunity evolved 450 million years ago. They are saying no. I showed you the one graph. 700 million years ago, innate immunity. 450 years million ago, adaptive immunity. Then we are having both innate and adaptive immunity. So this we have seen it in the sponge. This we have seen it in jawed fish. And this is human. So which means innate immunity evolved 450 million years ago. That is wrong, which is around 700 million years ago. And adaptive immunity system evolved in 450. So they asked are wrong. So
no problem.
1 and 2 are wrong. Innate immune genes evolve slowly while pathogen evolve rapidly. Yeah, that is also that we have seen one pattern how pathogen will go faster and innate immune genes are evolve slowly. The reason is very simple. These are non specific. There's no specificity for innate immunity to act. So it can act on multiple things. Anything can happen. So final thing is, which of the following statement is or correct? Uh, Lysosome destroys the cell wall of bacteria. Yes, that is correct statement. Dermis gives the skin strength and pliability to resist abrasion that could introduce microorganism. That is also correct. And sebum increases the pH of the skin to a level inhibitory to many bacteria. No, it will reduce the pH. It will become acidic. So this is the wrong option. So 1 and 2 are correct. So if you see the overall today, we started with the recap on fifth week concepts. We talked about the microbial community inside resist inside the body, that is called microbiome. So how much time it is high, that is around 10 times. Then we have compared with the human genome as well as the human microbiome actually. Then what are the different types of interaction between the microbes and then the host can occur, that include commensalism, mutualism and parasitism. Then how the evolution of innate as well as the adaptive immunity takes place, that we have seen. Then innate immunities are the first line defense of the human life, that also we have checked it. Then what will happen each and every step, barriers are there, whether it is a physical or the anatomical barrier or the mucous barrier or the effector cells will be there or the recognition receptors will be there. What will happen if each one get fail, that we have seen. Then we have majorly talked about innate immunity. Then we talked about the mechanical barriers. Then skin we talked about elaborately. Then the mucosal surface, including the bronchial ciliated epithelium, gut epithelium that we have seen. Here we have talked about one big cell, that is a Pannett cells, which can secrete the this defensins, and then the reg3, and then the lysosome we talked, and then which will majorly digest the bacterial cell wall. Then we talked about the how these pre-pro forms or the inactive forms of defensins or the antimicrobial peptides will be synthesized. Then what are defenses we have seen, and other antimicrobial peptides we have seen. Then the biochemical barriers, including the reactive nitrogen species and then the reactive oxygen species we have seen, and we have seen about the microbiological barrier. Then we come to see that what is there any kind of relationship between the innate and then adaptive immunity. Then people started seeing it in the Drosophila. Then they started seeing it in the mammalian system, including the mice. They have seen.
Yes, sir.
Then what are the systems in innate immunity response of fungi we have seen. Then we talked about Professor Akira's
Yes, sir.
work in this TLR pathway. Then finally, what is the overall thing is happening. Then finally we compare with the human thing. So in the human PRRs, it can be TLR, RLR, and then NLR will be there. Then how we talked about
Okay,
TLR
sir.
majorly, it can exist in the membrane form, and then it can be in the endosome
Yes,
form
sir.
as well. So what are things will be directed by this TLRs we have checked. Then their resident, or what are the cells expressing TLRs that we have seen. Then TLR's activity we have seen, it can be the MID88
Bye,
dependent
sir.
or
Good night.
MID88 independent pathway, which is the TRIF dependent pathway. Then
Yes,
finally,
sir.
what is happening to the pathway once
Okay,
get
sir. Bye, sir.
activated?
Thank you,
Then
sir.
we
Good
talked
night,
about
sir.
the type 1 interferon activity. Then the different polymorphism, how it reacts in terms of TLR2, 4, 5, then MAL, IRF5, IRAK4, and MID88. Then we talked about this great scientist, that is Kohli, how he introduced this streptococcal things into the patients. It's called toxins. Then other drug molecule or the this bacterial products in the drug testing, what are things are there. Then how they are using it for the other class of drugs. So then we have seen other around 10 questions for the day. Then with that we are finishing the week 5 recap. Understood? Do you have any questions to ask? Oh, it's happening like this. No, I'm really sorry. Something happened. It went. Which question it stopped actually? Subbalakshmi?
Sir?
Which question?
Seventh question.
Sorry.
Seventh question.
Okay, okay. I will repeat it. Sorry. Okay. Seventh question. You stated this thing. I don't know some issue. Oh, eighth question. Which among the following statement is are correct? They have asked. So they said
Your
microbe.
screen is audible, sir.
What?
Screen. Your
Oh,
screen
yeah, yeah,
is
yeah,
not.
yeah, yeah. 2 minutes. I'm really sorry. Here there is some network error. That's why.
Okay, sir. No problem, sir.
Yes. Is it visible? So seventh
Yeah,
question
yes,
is
sir.
bacteria and bacterial products. Then we came down to this eighth one. One more person is asking. Let me- Yeah. Eighth question. Microbes are 10 times more than the number of human cells. So that is a correct statement. Yes. And every day an adult human swallow digest about 1 liter of mucus. That is also correct. The answer wrong is microbiome present in the human body does not include fungi. That is a wrong statement because microbiome means all microbial things, na virus, bacteria, fungi, algae, everything. These things are not there, but I'm talking about including all. Then, which of the following statements regarding innate immunities are wrong? They asked. So, innate immunity evolved 450 years. They have 450 million years. They said. That's what I told. Now we were talking about one thing. We have seen that 700 million years ago, innate immunity came in sponge. Then adaptive immunity came 450 million years ago, and we are having both innate and adaptive immunity. Okay, so that's why first two statements are correct. They are wrong actually because they have given innate immunity 450, it should be 700, and this should be 450. Then finally, innate immune genes evolve slowly while pathogens evolve rapidly. So this is a correct statement, but the question asked about wrong statement. So option A and option B are correct options for this question. Then finally, which of the following statements is are correct? They ask. Lysosome destroy the cell wall of bacteria. Yes, that is correct. Then dermis gives skin strength and then pliability to resist the abrasions that could introduce microorganism. Yeah, that is also correct. Then finally, sebum increases the pH of the skin. So this is a wrong. Sebum will reduces the pH. So that is the correct option. So far the whole day that we have talked about this day, we have started with innate immunity. Then we are talked about the barriers of the body. Okay, physical, mechanical, microbial, and finally we have seen about biochemical. And finally we saw that what is the relationship between innate immunity as well as the adaptive immunity. It started with Drosophila, and people have checked it in the human as well. And we talked about cholestoxin, how that will be helpful in the tumor research. So that is what we stopped it today. Understood?
Yes, sir.
Yeah, overall done. So you want to ask any questions?
No, sir.
Okay. Anyhow, whatever I missed it, what I did after that, I have given one recap of everything, what we have done the whole lecture today. That will be there in the recorded lecture, so you can go back and then check it if you want. Okay.
Okay, sir.
So we'll see you tomorrow. So see you next week. Okay.
Yes, sir.
27,
Okay.
27, not first. That next week when is coming? First, if I'm not wrong. Right? First. We'll see. Okay. Bye bye. You
Bye,
can
sir.
mail
Good
me if
night.
you want any details about that internship or something. Okay. Anytime. I will reply. I have given you
No,
mail ID
sir.
right already. Yeah. Take care. Bye bye.
Okay, sir. Bye,
Bye
sir.
bye.
Thank you, sir. Good
Thank
night, sir.
you. Good. Bye bye.
Bye.